All right, hello everyone and welcome. My name is Alyssa Creamer and I am the Director of Community Services at the Immunodeficiency Foundation, and I would like to welcome you to this education session on Wiskott-Aldrich syndrome. The Wiskott-Aldrich syndrome, precision medicine, where are we now? What does the future hold? We have a terrific panel of presenters today to share information about Wiskott-Aldrich, and you'll have the opportunity to ask them questions at the end. So at this time, please join me in welcoming our presenters, Dr. Fabio Candotti from Lausanne University Hospital and Lausanne University in Lausanne, Switzerland, Dr. Donald Kohn from the University of California, Los Angeles, and Dr. Matthew Porteus from the Stanford School of Medicine.
Okay. So thank you very much for this opportunity to talk about Wiskott-Aldrich syndrome. I actually did my fellowship with Michael Blaze at the NIH 40 years ago or something, and saw more Wiskott patients in that 2-year period than I've probably seen in my career since, but it's been a long-standing interest because of all the patients and families I met at the time. So this is my conflict of interest. And the relevant one is there's a startup company called Immunovec, and we've made a new lentiviral vector for Wiskott-Aldrich syndrome that I'll talk about at the end. And I am on their SAB, and they've licensed the intellectual property from my university, in which I'm an inventor. So I have financial conflict of interest. But hopefully it won't change what I say. So just to start at the beginning, because this is sort of the introduction for both my talk and for Matt's. As we know, Wiskott-Aldrich syndrome can be essentially cured with a bone marrow transplant. And when we take bone marrow or blood stem cells from a patient, we get the whole family of cells, but it's really the stem cells in the bone marrow, which are 1 in 10,000 or less, that are the permanent cells that in the new patient will make their blood cells forever. So to do gene therapy, that's the cell that we have to fix. And so we sort of have 2 ways of doing that. And I will talk about adding a copy of the normal WASp gene to the stem cells. Then Matt will talk about actually fixing the WASp gene in the stem cells with CRISPR approaches. And so both methods kind of have a bit of a common platform. And the idea is for gene therapy, it's autologous, meaning we use the patient's own stem cells. So we collect the stem cells from the Wiskott patient in this case, either from a bone marrow harvest or by mobilizing them into the blood and collecting them. And then we can take those stem cells to the laboratory and do the genetic correction. So again, I'll talk about using viruses that will integrate a normal WASp gene into the stem cell. Matt's going to talk about using CRISPR and other approaches to actually fix the endogenous WASp gene in the stem cells. And then usually before they're given back, the patient will get some chemotherapy to condition their marrow to make space. So then the cells are given back intravenously. They'll make their way back to that now emptied out bone marrow and take up residency and start making all the blood cells and especially now they'll be able to express the normal WASp gene and do all the things they weren't doing before. And so for the viral approach, that's been the classical approach now. There's a family of viruses called retroviruses, and there's sort of a submember of that family called lentiviral vectors or lentiviruses, and they both have the same biology. They will take their genetic information, which actually in the virus is RNA, copy it into DNA, and then that integrates into the chromosome of the stem cell. And so if you've cloned a normal WASp gene into the virus genes, they'll integrate a normal copy of the WASp gene into the stem cells, and that will then get passed on to all the billions of blood cells that stem cell makes. They'll inherit now this normal WASp gene, and if it expresses and makes WASP protein, the cells can function normally. And so the first trials for gene therapy for WASP use sort of version 1.0 of delivery, which were mouse-derived retroviral vectors, sometimes called gamma retroviral vectors. So a group in Europe used a retroviral vector with a very strong promoter to drive a lot of WASP protein. And when they treated patients, they reported initially this led to very good restoration of immunity and platelet counts after they transplanted back into the patients their own stem cells that had been corrected with this virus. However, they had really severe complications, and at least 7 out of the 10 patients they reported develop basically leukemia from the virus. So that's obviously, an unacceptable complication. And this is just from their paper they reported. These mouse retroviruses have very strong, what are called enhancers, which can drive expression of the gene. So it can make a lot of WASp protein, but those enhancers can turn on a nearby gene where they've gone into the chromosome. And so if they've happened to land next to a gene that tells the cell to grow, and they stick that gene in a stuck-on position, it can make the cells grow out. And this is a blood smear showing many, many white blood cells that have large nuclei that are leukemia cells. And so that has been seen, this retroviruses causing leukemia has been seen first in X-linked SCID, here in Wiskott, and was also seen in CGD. And so the field has really much moved away from these mouse retroviruses to the second version, which are the lentiviral vectors. And these two types of viruses are cartooned here. So what I talked about, the mouse retroviruses that have these strong enhancers, they can turn on a nearby gene where they've landed. So if you put genes into 10 million stem cells, each cell is going to have it in a different place. The ones that happen to be next to a growth gene and turn it on can cause that cell to grow out of control. And so sort of in the early 2000s, the second generation of vectors were made, and either used with a retro, but mainly with lentis, where those enhancer elements are deleted. So you can't turn on a nearby gene, and you just run the gene, like the WASp gene, off an internal promoter. And so these are the types of vectors that have been used now in the trials really since about 2000. And they've proven to be much safer, and no one has developed those complications that were seen with the mouse retroviral vectors. And so, in fact, there were 4 trials using the same viral vector that I'll talk about, 3 in Europe and 1 in the US, use the lentiviral vector. It has a normal WASp gene, and it's driven actually by the promoter from the WASp gene itself. And the thought is, if that's the promoter that tells the gene normally when to be on, it should work well in the vector. And sort of, I'll go into more details of what the study showed, but just as a summary, they showed good immune reconstitution, I think, in all the patients, but only a moderate improvement in platelet counts. But there have been no toxicities. And between the 4 studies I'll show you, I think 34 patients have been treated that are out now 5 to 10 years, and there have been no complications from the vector using these second generation lentiviral vectors. And so this is, on the left, is a cartoon of the vector. And so these are the viral elements, but it has a normal human WASp gene. So that makes the WASp protein that's missing, and it runs off the WASp gene promoter. So it used the promoter from the WASp gene to drive the normal gene. And the vector was sort of developed as the EU consortium between Milan, Italy, France, and London. And then multiple trials were done. And this is from one of the trials just showing the lead up to it. And so, gene therapy is a bone marrow transplant. It just uses the patient's own cells. So typically some chemotherapy needs to be given first. So this was the schema for one of the trials where they collected bone marrow. And in this earlier study, they were giving the cells back fresh. Like they collected the bone marrow on Monday, and during the week it was in culture to add the gene with the virus. During that time, the patient got 2 chemotherapy drugs, busulfan and fludarabine, to make space for the cells to come back in. And then on Friday, the cells that had just gotten the gene put into them were given back to the patient. More recently, most trials are actually, after the end of the gene transfer, are freezing the cells so they can be tested and given back a few weeks later. But earlier, most trials were given the cells fresh out of culture. And so this slide summarizes those 4 trials. So between London, Paris, Boston, and Milan, 34 Wiskott patients received that lentiviral vector. None of them needed a transplant afterwards, meaning it didn't fail to reconstitute their immunity. And there have been no reports of leukemia or any other problems from the vectors at the present time. And so a little more details about the trial. I don't know which trial this is from, but it's one of the early trials. And they were looking over 2 years for outcomes. And they wanted to see improvement in something meaningful to the patient. So did their eczema get better? Was their bruising and bleeding better? And did they have less autoimmune complications? So those are obviously important to the patient to show improvement. So it was for males because Wiskott-Aldrich almost exclusively affects males. They had to have severe disease based on the score and or showing that they didn't express any WASp protein. You needed to show that the gene had the mutation. If they had a matched sibling or matched unrelated donor, they were excluded from the early trials because we didn't know if the gene therapy would be safe and effective. And if they had had a prior transplant that failed, they were not eligible to get gene therapy. So this is a slide of data from the London group from Adrian Thrasher. This is flow cytometry looking at expression of the WASp protein. So the cells are actually stained with an antibody to the WASp protein. And across the bottom are the healthy donor cells. So going to the right is brighter fluorescent. And so the open curve is no antibody, and that's the background. And this is staining with the antibody to WASp. So you see that all these different cell populations, granulocytes, T cells, B cells, and NK cells, are expressing WASp. So this is one of the patients from London at 12 and 24 months after gene therapy. And you can see that they are now expressing the WASp protein again in all these cells. You may also notice that it's actually not as bright. So if you look at what the expression pattern looks like in the patient cells, like we'll take the T cells here, it's a very sharp, narrow peak, meaning most of the cells are expressing the same level. Whereas with the gene therapy, it's not as bright, it's not as far to the right, and it's actually broader because when you put the vector in, every place that it lands may influence how it expresses. So expression is somewhat lower and somewhat more heterogeneous. And that's the biology of the vector that was learned from these studies. And the major limitation was in platelet recovery. So this is a slide from a paper from the French group reporting platelet counts in all these panels from 7 of the patients they treated. And so you can see, normal in their lab is above 110,000. You can see that most of the patients' platelet counts did not recover to that point. This one patient had a splenectomy and their counts came up. But the other ones really only had 20 to 40,000 platelets. So they didn't really show much improvement in platelet counts. And this is the French cohort. And they looked at how many cells, how many of the patient's own stem cells with the vector were given back at the time of the transplant. And those patients that got more stem cells, like 10 million per kilo, tended to have higher platelet counts. So more cells transplanted seems to help that problem. This is the Milan group showing that their patients' platelet counts went from about 20,000 to 40,000 or so. So it was an improvement, and 40 is probably better than 20 for bleeding risk, but it's not normal. And they also measured how many bleeding episodes the patients had after gene therapy, and it was definitely reduced. So this helped the patients. It didn't normalize things, but it helped their bleeding problem. And then this is from the Boston group, and Sung Young Pai shared these slides with me. So this is measured, one of the endpoints we often look at gene therapy, how many cells in the blood have the vector, or what's the vector copy number? So vector copy number of 1 is like every cell has 1 copy. And so what you can see is the patients sort of fell into 2 groups, those with low number of gene-corrected cells in the blood and those with higher. And when they looked back at the stem cells that they had treated with the gene before they gave them back, sort of the graft, these 2 had more copies of the vector in the stem cells that went into the patient than those. So it says, so one graph said more cells is better. This is saying more copies of this vector is better. And then when they looked also then at platelet count, in fact, those 2 that had more vector in their stem cells and more cells in the blood with the stem cells are the ones that had the better platelet counts. And so the implication of this is that you needed 2 or 3 copies of the vector per cell to make enough WASp protein to really normalize platelets. So this, this is not the ideal result, but it tells you sort of where we are. So our group actually, and this is the work that we've licensed to a company, went about trying to make a better vector to express better in platelets. So our goal was to increase expression in platelet, the cells that make platelets, megakaryocytes, compared to the vector that I just showed you, which we're calling the WASp 1.6, while maintaining good levels in the lymphocytes that seem to get better with that other lentiviral vector. And we know, I said early on that the retroviral vector, although it was toxic, it restored platelet counts. So we use its level of expression sort of as our bar. And the approach we looked at, so this is a little complicated, but I'll explain it. So this is sort of a gene map of the chromosome region where the WASp gene lives. So there's the WASp gene itself, and these are all the DNA around it. And there are ways to look at what other elements around the gene might turn it on to express better. So it's not all contained right here. And so my lab has developed a method to look at all the potential sequences that might turn the gene on more to a higher level from its own vicinity. And so this is then looking at expression from a whole series of vectors where we've added those different elements on top of what was in the original vector. And a couple of those elements added from the WASp gene increase the level of expression. So by combining those together, we made a vector, and this is again showing expression level in platelet precursors. So our modified WASp vector expressed as well as the retroviral vector did, and higher than the lentiviral vector that's been used in the previous studies. So this is just laboratory correlation. We hope to see in a clinical trial that we get better restoration of platelets with only a single copy of the vector per cell. And so that's my group who did the work. And I just want to point out Ryan Wong, who's standing next to me here, did all the work making this new WASp vector that hopefully we'll have in clinic in a year or so. And I will stop there. Thank you.
Okay, I'm next. My name's Matthew Porteus. I'm a physician scientist at Stanford, trained in pediatric hematology and oncology, and I'm pleased to be able to follow Don and participate in this section. And what I'm going to talk about is what people call genome editing as the next generation of gene therapy. And the idea in gene therapy is, of course, to give back a gene to the patient's own cells and to correct the underlying defect and make the person healthier. And Don really nicely described how using viral vectors to deliver a good copy of the gene to the patient's own cells can improve many of the symptoms of patients with Wiskott-Aldrich syndrome. But he also highlighted that not everything was improved. And one of the things about gene editing is to think about how we can improve on that really, really great start. I also have some conflicts of interest, potential conflicts of interest, although none are relevant to Wiskott-Aldrich syndrome. And as many of you know, of course, diseases like Wiskott-Aldrich syndrome, there's thousands of them affecting hundreds of millions of people around the world. And again, this is an audience that already recognizes that it's not just the patient, but it's the community around that individual that's affected by the disease, what I sometimes call the echo of the disease. And so the goal of all of us is we believe that if we can cure that one patient, we not only make that individual better, but we can make the community around that patient healthier. There are many genes involved in genetic primary immunodeficiencies. And a lot of these need the expression of the protein from that gene at just the right amount in just the right cell at just the right time. And while Don described how we can express the Wiskott-Aldrich protein gene using a lentiviral vector with a promoter, what was clear from the clinical trial is we were getting some amount of protein in the right cell at more or less the right time, but it wasn't perfect. And so the idea with gene editing is can we achieve that ideal goal of the right amount in the right cell at the right time? And the way we do genome editing for a disease like Wiskott-Aldrich syndrome follows this basic structure, which is we design a specialized protein called a nuclease. And a nuclease, this specialized nuclease is designed to bind, once it enters the cell, it finds a specific sequence in the genome and makes a break there. It basically damages the DNA. And this damaged DNA, this broken strand of DNA, can be repaired in one of two major ways. One is by what is shown over here, non-homologous end joining, in which the two ends are just stitched back together. And for a genetic disease like Wiskott-Aldrich syndrome, that stitching back together really does nothing to improve the gene or fix the gene. So we have to harness the other way that cells, the other primary way cells fix breaks, which is called homologous recombination. And in homologous recombination, the machinery looks for a piece of undamaged DNA that has similarity to the site of the break. And it will make a copy of this undamaged DNA and paste that copy into the break. And so by designing what we call the donor DNA, we can trick the cell into fixing the break and making either single nucleotide change, single changes in the letters of the DNA, or actually inserting a full gene into the DNA at a precise location. So in contrast to the viral methods, now we can have the potential of putting the good copy of the Wiskott-Aldrich gene exactly where we want. Now, the way we do this is as Don described, it's an ex vivo process, meaning we take the cells outside the body and we genetically engineer them in the laboratory. To do genome editing, we use the CRISPR system, and we use it in a very specialized manner where we take the Cas9 protein, which is the molecular scissors that makes the break, and we complex that to a guide RNA, which tells the molecular scissors where to make the break. And we deliver this combination of our protein and an RNA molecule into our purified stem cells via electroporation, a method of delivering proteins into the cell. But we also need to deliver that correction DNA, that donor DNA that the recombination machinery needs to use. And for that, we actually repurpose a virus called adeno-associated virus, which has evolved to deliver its DNA to the nucleus of cells without being detected. Our cells are well designed to recognize viral infections. But in contrast to the viruses that Don described, this is a virus that does not naturally integrate its DNA. So in a dividing stem cell, what would happen is the AAV would get in, and then over time, it would just be diluted out and dissipate and degraded and would no longer be there. So it is a way of delivering the donor DNA in what we sometimes call a hit and run method, where it just needs to be there for a very short period of time, serve as the template for the change we want, and then it can go away. Okay, so the first disease I'm going to talk about is not Wiskott-Aldrich syndrome, but is a very related disease called severe combined immunodeficiency X-linked. And this disease, like Wiskott-Aldrich syndrome, has mutations that are throughout the gene that can cause the disease. And so what we wanted to design is a method that could functionally correct all of these mutations at once without having to go through and design a specific method for every single mutation. And even which would be technically possible, but very time consuming. And from a standpoint of bringing each process to patients, very cumbersome. So before I get into that method, again, just reiterate that the key cell to correct here is the hematopoietic stem cell, because ultimately it gives rise to the lymphocytes that are affected in Wiskott-Aldrich syndrome, the platelets that are affected, and even the macrophages and monocytes that are infected. So by correcting this stem cell, we can give rise to functionally corrected blood cells in all the different lineages. And so the approach that we have adopted for SCID-X1, and I'll describe later how it's been used for Wiskott-Aldrich syndrome, is like the gene therapy approach that Don described, in which we take a good copy of the gene and we put it back in the genome. But instead of putting it back into the genome in a place we don't control, we use genome editing to put it back into its own gene. So we find the start of the gene and we put a good copy right at the start, so that all of the downstream mutations are no longer read by the machinery that turns genes into proteins. And in this way, because we are putting it where it naturally lives, all of those complicated regulatory interactions that Don described, we don't even have to learn about. We just hope that by doing this, we've captured all of the natural regulation so that the gene will now be expressed, as I said, at the right cell at the right time in the right amounts. And for SCID-X1, we fundamentally showed that this worked. We could get rescue of the amount of signaling through the cell by putting the gene, wild type copy of the gene, back into itself. And one of the interesting studies was done actually by our collaborators at the NIH, Harry Malik and Succeed Raven, who have looked at gene therapy for SCIDX1, as well as collaborated us on genome editing for SCIDX1. And what they showed is that actually the development of NK cells, a specialized immune cell that is defective in SCIDX1, was better rescued using the AAV approach, the gene editing approach, than using the lentiviral approach, where you can see there was very little development of NK cells using the lentivirus compared to the rather robust development of NK cells using gene editing, pointing again to the idea that gene editing, because you're putting the gene back into itself, you might get better functional outcomes. And we showed that these genetically edited cells still retained their stem cell characteristics as much as we could determine before testing this in patients. So now last year, a very similar and exciting paper was published from Adrian Thrasher and Alessia Cavazza, applying the strategy to Wiskott-Aldrich syndrome. And the idea is that they would knock in either a optimized version of the Wiskott-Aldrich syndrome gene into its natural locus, and they had some bells and whistles, and they wanted to compare two versions. And what was very exciting about this paper is they compared the gene editing knock-in approach to the lentiviral gene therapy approach that Don just described using the 1.6 vector he mentioned. And what they found is that both methods could reliably create cells that express the Wiskott-Aldrich protein. So here in figure D, you can see using the gene editing, about 40% of the cells or 50% of the cells express the protein. And with the virus, it was still a good percentage, maybe not quite as high. And this was reproducible across stem cells from 4 different Wiskott-Aldrich syndrome patients. But then what they did is they compared, well, what happens to the function of the cells derived from these genetically engineered cells? That is, how well do the macrophages work when you use gene editing versus lentivirus? And what they showed is that if they looked at the amount of Wiskott-Aldrich syndrome protein made in the macrophages, it was close to 100% of the normal levels, whereas in the lentivirus, it was about 80% of the normal levels. And when they looked at whether the macrophages made something called a structure of the cells that we call potosomes, they found that compared to wild type cells, which would be 100% of normal, the gene edited cells were about 70% of normal compared to about 40 or 50% of normal using the lentiviral vector. So an improvement, although not complete normalization of a specific function of macrophages that is caused by defects in the Wiskott-Aldrich syndrome gene. And then Don mentioned that in the gene therapy trials, while there was excellent T cell function, that there was still challenges with generating enough a normal platelet number. And so again, what Dr. Cavazza and Thrasher did is they compared how well Wiskott-Aldrich syndrome protein was made in the megakaryocytes, the cells that will give rise to platelets. And they found that using the knock-in approach, there was near complete normalization of the protein level in the megakaryocytes compared to only about 40% of the necessary or of the normal level using the lentiviral vector. And then they looked at different features of the platelets that were derived in terms of size, how granular they are, whether they were inappropriately activated, and whether they were undergoing cell death inappropriately. And again, using all of these functional assays of platelets, they showed that the red was better than the purple, and the red was almost equivalent to wild type cells, so as if the cells were completely corrected. So what I list here is just broadly that this has been now done across a number of different immunodeficiency diseases, including Wiskott-Aldrich syndrome, to modify the Wiskott-Aldrich syndrome gene. But I will note that this is still all preclinical, meaning it is all work that's done in a laboratory and work that's done in animals and has not yet been tested in patients. But the preclinical data, I think, supports that this is a method of potentially treating and improving the lives of patients with Wiskott-Aldrich syndrome that might even work a touch better than the lentiviral approach that Don just described. And I think Don and Fabio and myself all believe that it is important to have as many shots on goal and as many opportunities to improve the lives of patients and not put all our eggs in one basket. And I think we're all excited that this new approach hopefully can reach the clinic in the near future. So again, thank you for your attention and the opportunity to present the work. Hopefully I didn't present this in a too complicated or technical way, but I'm looking forward to the discussion that we have following. Again, thank you very much.
Wonderful. Thank you, Dr. Kohn and Dr. Porteus. I am now going to hand it over to Dr. Candotti, who's going to manage our wonderful Q&A session and the discussion between Dr. Porteus and Dr. Kohn. So Dr. Candotti, I believe you have all the questions that have been asked so far, so I will let you take it away.
So thank you, Don. Thank you, Matt, for those presentations. And to the audience, you've heard about two approaches of precision medicine, meaning to medical approaches that are specifically designed to treat one specific disease, in this case, the Wiskott-Aldrich syndrome. And I'm going to start with Don. And for people that don't know, Don is not only a gene therapist, but also is a bone marrow transplanter. So he has worked with both approaches. And so I'm going to put him on the spot right away and ask him, what is better? Is even possible to ask that question? Are there enough data from these two different approaches to being able to counsel patients? Because at this point, for gene editing is not yet the case, but many situations, families are faced with the two options. And can you compare and contrast for us, please?
Yes. So I agree. Your statement that we don't have enough numbers yet to say anything is true. As I showed with the Lenti, at least when that group was compiled, was 34 patients, which is a small number. The potential advantages of gene therapy, whether it's with a lentiviral vector or editing, as Matt was talking about, is that it's the patient's own cells. So in theory, they should not be rejected like another person's cells could be. And more importantly, from transplant, they shouldn't attack the patient. And so for those of you who've had experience with bone marrow transplants, you know that one of the real problems can be graft versus host disease, where T cells from the donor that come along with the stem cells can react against the patient who gets them. And so that's if you have an unmatched donor, that's going to be very severe. If you have a well-matched brother or sister, it's less likely to occur. And in between, sort of a well-matched unrelated donor has sort of a middle risk of graft versus host disease. And GVHD can be mild, or in some cases, it can be quite severe, quite long-lasting. And so 10 to 20% of patients who get a transplant can have mild to severe problems with graft versus host disease. And the hope for gene therapy is that we'll have none of that. And certainly with ADA SCID, where we've transplanted 60 or so patients with gene therapy, we've had zero GVHD. And also, the other potential advantage for gene therapy is that we probably can give less chemotherapy and less immune suppression than you need to get acceptance of a graft from another person. So the transplant should be somewhat milder. The risks of complications should be somewhat lower. And so if gene therapy works, A, and B, if gene therapy is available, which is also another major bottleneck currently. I think it should be better, but we can't say that yet with the numbers that are treated. And I show that we maybe have a better lenti. Matt showed the potential advantages of gene editing. So unfortunately, we may not know the absolute answer to that for 5 to 10 years until those methods are employed and patients are followed for some time to know what the outcomes are.
Right. I'm going to add to this discussion a question from the chat, and which is about if you can talk about the mortality that has been observed in gene therapy trial, and what will be the efficacy for adult patients, perhaps patients that have autoimmunity.
Yeah. So in terms of mortality in the trials, I basically showed what data there are. So with the retroviral vectors, 7 of 10 developed leukemia. I actually don't know how many of those were, cured of their leukemia by treatment versus how many of those may have died from that complication. So I'm not sure that number. With the lenti, of the 34 patients between those 4 studies, all of them are alive. So there's been no deaths in the Wiskott gene therapies with the lenti. Small number. One of the other things that somewhat biases, often patients to be eligible for a gene therapy trial need to be in perfect health and not have infections, not have problems, where sometimes a bone marrow transplant is done despite those problems just to try and get a therapy into the patient. So there may be some bias taking healthier patients to gene therapy. And so again, it's the kind of thing that we really need further clinical experience to know if all things are equal, is gene therapy going to be better than a transplant from a donor?
Right. For Matt, a question from the chat that says, could you please explain the complexity of using gene editing for long genes such as WASp, and where there are at least 300 different mutations known?
Yeah, it's a great question. And clearly I went over that part a little too quickly. So a couple things, and it relates to another question about the ability to put in the full replacement of the gene. I'll take a couple minutes. So all genes, including the Wiskott-Aldrich syndrome gene, have what we call exons, which are the part that get converted into a protein, and the introns that are between the exons. And when we put in the new gene, we take out the introns and we just put together the exons so it becomes much, much smaller. And that is why we can fit that into our vector and have it insert. So it allows us to fit this really large gene in the genome into a much smaller package that we can package. And that's what's done in the lentiviral gene therapy as well. The issue of the 300 different mutations is really right. And so that is why the field is looking at this way of not trying to design individual corrections for each mutation, but instead take the good copy of the gene and put it right at the very front of the gene, right at the start of the gene. And so now when the cell reads the gene, it reads the good gene, and then it stops reading because there's a signal that says, this is the end of the book, and you're done. What that leaves is the mutations that are left behind never get read. They are never, the cell never sees those mutations. And so by inserting the good copy at the front, we functionally can correct all of the different mutations because functionally they all get put into a part of the genome that never gets read. So hopefully that explains the question about how we can do one approach to correct many different mutations at once.
Right. And that also explains, probably has given the answer to another question that said, is that the full replacement of the gene, the original gene stays? Is that a problem that the mutated genes remains?
Yeah, and I think the mutated gene remains, but we have designed the gene editing such that the mutated gene never gets read anymore. So it's essentially, it's there, it's present, but it's silent.
Exactly, right. So Don, there are still questions about comparing bone marrow transplant and gene therapy, and also about the decision-making process, of that the family needs to take when they have those options. One specific question is if a patient has a sibling match, would you recommend a BMT or to try gene therapy first?
Yes. So, in general, the transplant results are much better if there's a matched sibling to be the donor than if you have to go outside the family. And even a perfect match outside the family isn't as close genetically to the patient as a sibling would be. And so most of the gene therapy trials to date, to be conservative, have not allowed people with a matched donor to be in the study. Felt that because the matched donor is sort of the standard of care, that they should have that, and that initial trials of gene therapy with unknown benefit, unknown risk, would be for those that didn't have a good match. For ADA SCID, we've been saying now that the results have been so good with gene therapy that it's sort of the family's choice whether that they're roughly equivalent outcomes. And I think with a little more work in gene therapy and sort of these next sets of approaches, it may be as good. And even with a matched sibling donor, there can be graft versus host disease in 10% of patients or something. So in theory, one day gene therapy may be known to be better, but it's a complicated decision because the amount of information is limited. And again, as I said before, the other problem is having gene therapy studies that are open. Again, with ADA SCID, one of the problems we've run into is we've kind of now proven that it works. So it's hard to get a research grant to do another study because it's no longer a research question. And so until it then gets taken by a company and made into an FDA-approved drug and sold, there could be a many-year window in which gene therapy is not available just because it's no longer research, but it's not yet a drug. And so I don't know if there are currently any open trials of gene therapy for Wiskott. I don't know if any of the trials are currently enrolling. I think all 4 sites kind of did their number of patients. And, so I'm not sure until the next generation of trials come along if gene therapy is going to be an option for the next year or two.
Right. So for gene therapy, Milan, the Milan group can how you say, present the cases to Orchard, and they have had a series of approvals. So there is still a center in which some patients are being treated.
So like on a compassionate basis?
Right. Right.
Okay.
So Matt, if you could look into the crystal ball, when is gene editing going to be available to the clinic? And at that time, will adults of 20, 30 years of age be eligible for that therapy?
So thank you for acknowledging that it's a crystal ball, and our crystal balls are always rather flawed. I'll take the second part first, which is that a 20 to 30-year-old, assuming that they are in relatively good health, because as Don mentioned, there's sort of a window where we don't want somebody to be too sick to get an experimental therapy. But in general, at least in the US, and I think it's true in Europe, that's the perfect age group. As long as the symptoms of autoimmunity and organ and everyone and person's doing well. When is a really good question. There is what some people call the valley of death between the beautiful studies that Adrian and his group did at London and the opening of a clinical trial. For some of the reasons that Don described, there is not a lot of great funding agencies that are willing to take on the multi-million-dollar process of converting a laboratory-based process that can be done, as I say, on a random Wednesday afternoon to a well-controlled process of engineering cells, enrolling patients on a clinical trial, having the patients monitored in an appropriate way. And so it depends on money, enthusiasm, and commitment among a lot of people to do this. I would say it's generally taking right now in a very committed process, somewhere between 2 and 4 years to go from, say, the study I presented to a clinical trial. Of course, we hope that as people become more efficient at that translational process, it might shorten. But I think it's not unreasonable to think that there's still a 2 to 4 year window to get from really exciting mouse data to a first in human phase 1/2 trial. Don, would you disagree, or do you think
I agree
Maybe
with you.
I'm too optimistic even.
Well, yeah, I was thinking 3 to 5 years rather than 2 to 4, but it's a difficult process. And so once, the data that Matt showed from the Thrasher lab, that's proof of principle. To open up a clinical trial in the US, the FDA wants to see you do that process on a patient dose number of cells, like 100 million cells, 3 times. And you've already worked out exactly what the criteria are to say you've edited enough and the cells are healthy. So there's a lot of sort of laboratory work that goes around to manufacturing the cells. The FDA looks at drugs all the time, and they treat these gene modified stem cells as a drug. And so there's a lot of, I think we're very happy that the FDA very carefully looks at how our medicines are manufactured, but it's a high bar to reach. And Matt and I are both very fortunate to be in California for many reasons.
Yeah.
But perhaps the biggest one is the California Institute for Regenerative Medicine, CIRM, the stem cell agency. They understand that's the process. So they fund their grants to fund those stages of the process, from proof of principle to getting to a trial to doing a trial. And even with those fundings, though, it's still 3 to 5 years, and it's a multiple grant applications. There's gaps. One grant is ended, and before you get to the next one, you might have 6 months to a year of a gap during which you have a payroll to meet. And so it's a challenging process, and it's still largely being done at academic medical centers like Stanford, like UCLA, like University of Lausanne. And we have some strengths and weaknesses. I think we're very good at doing. Matt and I are both bone marrow transplant doctors, so we know how to do the clinical part. But it's still, it's challenging. It's not perfectly rationally organized. And if we had infinite funds, we could really make it much more efficient. But working within the system, it's slower than we'd like it to be to bring a new thing from, oh, look how well it works in my lab bench, to can I treat patients with this safely and effectively and consistently?
Yeah. Right. Don, a question about your vector and how, if you can repeat a little bit how that differs from the AUT's vector.
Yes. So it sort of builds upon it. So it has kind of the same core promoter that the European vector, if I can call it that, uses to derive the Wiskott gene, plus additional elements we found near the Wiskott gene that turn it on more. So it's sort of built upon that as the basis, but it expresses more because it has these additional elements to drive the gene.
Okay, so you showed that it expresses more protein in the cells, and you expect that to be good for platelets production, but also for all the other immune functions that need WASp, not only platelets.
Right, and it's been tested in the WASp gene knockout mouse model and shows it corrects things. Unfortunately, the mouse model doesn't really have a platelet defect. It doesn't reproduce that human component, so you can't really determine that. And so that's why, ultimately we have to do phase 1 clinical trials and see how it performs in humans. We expect it will perform better than the vector that's been used before, but you never know that until you've proven it in a clinical trial.
Right. Okay, so there are some questions that are not really associated with the precision medicine, but I think we can discuss them anyway. There is a question from a Wiskott carrier who has heard that there are possibly some clinical research ongoing that concerns carrier females who are symptomatic. Is that something that you are aware of? I personally am not. If you're talking about therapeutic approaches for carrier females that are symptomatic. I don't think I've heard of specific research being going on there. There's clearly the need to acquire information of what is the spectrum of symptoms and problems that the Wiskott carrier females show, and that kind of research, fact-finding research, is going on with through survey mechanisms. So those are available. Those are being carried out, and there are more coming. And through the Wiskott-Aldrich Foundation and Sumati Iyengar, you can get information about that. I don't know if you guys want to add anything to the Wiskott carrier females topic.
No, you answered it exactly right. I think the first step is to try and get a survey of how much of a problem it is. The dogma is that carriers are asymptomatic, but we know that's not true. But I don't think we understand the full problem. And so I think the survey mechanism is a great way. If the more people who participate, the more information that will be obtained. And then if there are specific problems identified, then therapies could be thought about to treat those problems.
Right. A question about the gene editing and gene addition strategies. Is there a possibility that going through one of these processes that fails would make a following bone marrow transplant more difficult or more toxic?
If you want, Don, I'll take it first because yeah. As Don mentioned, and as I think this audience seems very sophisticated by the types of questions coming in, as part of either a standard bone marrow transplant or a gene therapy type transplant, you need to get chemotherapy to create space for the cells to come in. Now, in principle, if you got a autologous gene therapy transplant and it didn't take, there's nothing that would preclude you going to an allogeneic transplant. The challenge is you've already then been exposed to some high doses of chemotherapy that do cause some tissue damage. And so there is a little bit more risk for any person if they have to get high doses of chemotherapy twice instead of once. So I would say that would be the first thing I would say is it's not so much that a failed autologous transplant would make the allogeneic transplant not work. It's the problem of layering more and more chemotherapy on someone. I don't know, Don, if you want to expand on that.
No, yes, then I agree with that completely. Yes, and so then when faced with that situation, you have to decide, well, what's the proper time between the gene therapy to do that? And there's no hard data on that. I think transplanter will say at least 6 months, maybe a year. Maybe if the patient's better, doing okay, wait 2 years. But again, it's very much it's nothing that's been studied of how well patients would tolerate that second treatment of chemotherapy before a transplant from a donor.
And then, Don, these patients that undergo gene therapy, how does their follow-up look like? How frequently do they need to go for checkups? There are things that need to look out for.
So typically, the gene therapy trials follow the patients actively on study for 1 to 2 years. And during that period, it might be monthly for the first 3 months, and then every 3 months out to a year or two, during which there are sort of research studies. How many cells have the gene, or all the various things we showed you the data on. Those might occur every 3 months over the first 2 years. In the US, the FDA requires that anyone who has gene therapy be followed for at least 15 years, although they don't really say what you need to follow. And so what we would typically do is after the patients finish their 2 years on the research study, we offer the parents or the patient to enroll into our long-term follow-up study. And that entails really not a lot. So once or twice a year, for them to be seen by their home doctor, have an exam, have some simple blood tests. And then usually we'd request a sample sent to us that we'll measure, do they still have the gene present, and look at those things. So it's not a lot, but it's just the FDA wants to keep touch with patients after gene therapy to make sure there isn't a cluster of problems late. And really that hasn't been seen. I guess just one other point to make about the gene therapy to date, one of the concerns is that if you take the stem cells out of the body and you put them into culture and you add a virus or you edit them, will you take away their permanent ability to make blood cells? Will you make them not stem cells anymore? And at least with all the gene therapies to date, really once patients reach their level of gene-corrected blood cells in the first few months, it's really held steady. So there's been very few cases that I know of where it's sort of lost late. So it seems to be permanent, which is the goal, that it's a one-time treatment.
Right. I just want to make us mindful that we only have 3 minutes left, so questions will be fewer now. Matt, what is that you think your field of gene editing needs to achieve? If you had a magic wand, what would you really change in your lab to get things closer to the 2 years instead of the 3 years?
Yeah. Yeah, I would say, let me give two answers. One of the things I would love to see, it goes back to our earlier discussion of how do you go from the proof of principle in the lab to a phase 1 trial. I would love to see there be more robust infrastructure and pathways to accelerate that process. Again, I don't think I see any reason in the data that came from Dr. Strasher and Cavazza why this shouldn't be tested in humans. It seems a shame that we're going to have to wait so long to see the definitive results in the first few patients. So I'd love to see a more robust pathway for people. I would say if I had a magic wand, the other thing that I would love to see is for us to be able to get rid of the chemotherapy that's needed to prepare the patients to receive these cells. Certainly for patients with severe diseases, including Wiskott-Aldrich syndrome, the risk-benefit or benefit-risk says high doses of chemotherapy is probably worth the risk. But I think we would all love to take that risk out of the equation. I think it would open up the use of these therapies to more patients, even in severe diseases, and certainly for more diseases that currently we wouldn't consider. So that's a magic scientific wand. And then I wish if I had my magic infrastructure wand, it would be to create a really much more streamlined process for all of us to take our laboratory discoveries and test them in the first few patients.
Forward, right. Don, we have 1 minute. And the question is, my son had a successful bone marrow transplant 3 years ago. As he gets older, would there be any reason to do gene therapy?
No, I mean, if he's had a successful bone marrow transplant, that he should be one and done and not need it any further. So if he got enough donor cells engrafted to make a good immune system, to make good platelets, that should last him his whole life.
Yep. And Don's former mentor and my current mentor did the first transplant 50 years ago, I understand, for a disease for this disease, right?
Right. In 1971, it was. Yeah, yep. Exactly 50 years.
So.
Okay, so I think we are out of time, unfortunately. I want to thank you again, Don. Thank you, Matt. This was very interesting and fact filled. So people hopefully have gotten a good idea of where we are going with this precision medicine for Wiskott.
Thank you.
Alyssa.
Yes. Oh, thank you so much, Dr. Porteus, Dr. Kohn, and Dr. Candotti. I am so sorry to say that this session has come to an end. I wish we had unlimited amount of time to answer all of the questions and hear more from you all, but sadly we need to sign off now. So thank you again for volunteering your time to share your expertise with us today. I know the Wiskott-Aldrich community is so appreciative. So thank you again. And I look forward to seeing everyone at the next session that starts at 3:15 p.m. Eastern, swimming upstream, addressing payer challenges and ensuring access to genetic testing. So that should be an excellent session, and we hope to see you again shortly. So thank you again, everyone, for tuning in and for your wonderful questions, and for Dr. Candotti, Dr. Kohn, and Dr. Porteus one last time. And have a great rest of your day and your weekend, everyone. Take care.
Thank you.
Bye.
Bye, guys.